Hey everyone, this is Mohammed Salih from the Carpenter Lab at UCSC, and I'll be taking you through my project. In the Carpenter Lab, we study the innate immune response, which is your first line of defense against pathogen. Upon introduction of a pathogen, this response needs to be tightly regulated because too little of a response could result in inflammation, and too much of a response could result in overactivation of immune cells, such as macrophages, and overproduction of signaling molecules, inflammatory cytokines. For my project, I focus on heterogeneous nuclear ribonuclear protein, A2B1, which is a protein involved in transcript maturation and processing. In the lab, we have seen that in macrophages, which are first responders to infection, upon removal of this protein, there is dysregulation in inflammatory gene expression. This was our first piece of evidence to study A2B1's involvement in the inflammatory cascade. With that, I designed a mouse where this protein is removed only in macrophages. I extracted these macrophages and studied gene expression. I've introduced lipopolysaccharide, which is a bacterial ligand that activates inflammatory signaling in these cells. And I see that upon removal of A2B1, there is dysregulation in some key inflammatory genes. I've also introduced LPS directly into the mice and see that the knockout mice do not drop as much in temperature in comparison to wild type. They also have an impairment in cytokine production. I also studied a salmonella infection model where the knockout mice proved to be more susceptible and they died earlier. They also had a higher bacterial load in the spleen. With that, I've shown you data that A2B1 regulates the inflammatory cascade in the macrophage and in the mouse as a whole, and impacts survival outcome and cytokine production in response to salmonella. And I hypothesize that A2B1 is able to do so through regulation of transcript maturation and alternative splicing, which is where my project is going. And with that, I would like to thank my lab and all my funding sources, and especially the ArcSmart Foundation for its involvement and contribution to my professional development.